Okay, good morning everyone, and thank you first of all for the organizer for this extraordinary multidisciplinary exchange of ideas and for having me, and also to the previous speaker for setting the bar so high, no pressure at all. Today I will talk you about our latest results, about what we have been calling a game of tourism, that is gamma and beta oscillation in the basal ganglia. Now, why the basal ganglia? Very long story short, it is of great interest as a subcortical brain region due to Parkinson's disease. We are getting to know more and more of this subcortical brain region with the advent and widely spread use of deep brain stimulation technology. And we are developing this computational model of basal ganglia dynamics. Very long story short, it is subdivided. It is composed of thousands, more than 13,000 of spiking neural, leaky integrating fire, non-linear leaky integrating fire spiking neurons, subdivided into these three macro families. One is striatum, the external global pallidus, and the STN, the subthalamic nucleus. Each one of them is subdivided into subpopulation, shortly the fast spiking neuron, D1 and D2 receptor, dopamine receptor neurons within the striatum, and the GPTA and the GPTI in the external global pallidus, low discharge rate and negligible contribution for the former, and higher discharge rate and substantial striatal inputs for the GPTI. And then our STN, which is colored in red, it is our only glutamatergic nucleus within the basal ganglia. Now, this model was developed in our lab, not by me, but by my colleagues, in order to try to investigate beta rhythmogenesis. Beta oscillations, as you may know, are very widely investigated because you can we can find them consistently, and they correlate with the symptom severity in Parkinson's disease. You can find both in human subthalamic nucleus record, not only in the subthalamic nucleus, but in human recordings, and also in animal models of Parkinson's disease, especially in rats. And one figure I like by this Ortone et al. Plus computational biology paper is that, yes, this is the figure I was talking about. They were able to investigate the mechanism behind beta rhythmogenesis. Very long story short, they found that in low dopamine depletion state, that is the healthy scenario, they found the presence of two different beta oscillations regimes that are generated within these two nuclei that I try to highlight, the yellow and the orange one. And you can see that they are distinct in frequency. Each one do not interact with each other. But when you increase dopamine depletion, that is, you switch from healthy to the Parkinsonians, you can see that along the way in which this state, these dynamics, this computational model becomes pathologic, the two locks in, each one brings their energy together, and they match each other at a common beta frequency, and this is supposed the mechanism behind the beta hypersynchronicity. So one last introduction is about what is dopamine depletion, at least in this model. Dopamine depletion comes from in Parkinson's disease due to the nigrostriatal degeneration of dopaminergic neuron. And from the point of view of dynamics that we're interested in, experimental results are suggesting that it comes from an imbalance between D1 and D2 striatal population, these guys here, in which basically you can see that when dopamine depletion occurs, this guy here lowers the firing rate, while this guy here, D2, instead increases. And so from a computational viewpoint, the easiest way in order to replicate this experimental observation is to, in order to simulate dopamine depletion, increase the excitatory feed in the D2 subfamily. So, okay. So we have said that this model was developed to address beta oscillation. And beta oscillation comes from the interplay between these two loops. So it comes from the interaction across different nuclei. But the very same model displayed a very interesting observation for us that is also gamma oscillation, faster oscillation. And funny enough, it was, can I go back somehow? No, okay, it doesn't matter. It came from the two interesting nuclei. It is the D2, the one in which you inject the increased excitatory drive to simulate dopamine depletion, and the GPTI, which is the intersection between the two beta-generating subloops. And you can see there are some difference between the two gamma oscillations. First of all, top, the GPTI exhibit to display very fast gamma oscillation that are not that much interested about the Parkinsonian condition or the healthy one. And you can see also on the right that, of course, when we transition between healthy and Parkinson's disease, that is where we increase the dopamine depletion condition. We observe as a sanity check that beta power increases, as you observe experimentally, while in the globus pallidus, the gamma oscillations remain almost constant in power, when instead, the difference within the striatal, the two gamma oscillation, is that, of course, when we transition between healthy and Parkinson's disease state, we observe, again, as a sanity check, the increase in beta oscillation, and instead here gamma increases, and along the way between healthy and Parkinson's disease, it crosses the threshold of statistical significance. And you can see it here. In the healthy condition, there is no gamma oscillation, and instead we found it in the striatal gamma oscillation in the Parkinson's disease. So we have two different types of gamma oscillation, and we wanted to replicate what we have been doing for the beta oscillation in order to try to investigate the rhythmogenesis, the mechanism behind the rhythmogenesis of the gamma oscillation. So the first things we tried to investigate was the necessary condition for the gamma oscillation. In the plot in the left, that seems very complicated. Actually, it is not. What we did, we took every possible synaptic connection in our model and we dissected it. It is very complicated to do in experimental neuroscience, very easy to do in computational one. And we found that destroying the self-inhibition between the synapses between the globus pallidus, the one generating the gamma, with its neighboring neuron, eventually killed gamma oscillation. So we basically killed this inhibitory feedback loop here. And you can see also from the power spectrum of the firing dynamics of this nucleus. So in orange, you see the gamma oscillations in the original condition, and when we cut this self-inhibition, instead, you observe two phenomena. You observe, first of all, the disappearance of the gamma oscillations, and you can see that you have an increase of beta oscillations, so much so that eventually you see also a lot of harmonics in the power spectral density of the pharyngodynamics. And we also did something more refined, that's simply on and off of this synaptic loop, inhibitory feedback loop. That is, we try to modulate it along a continuum, but very long story short, we're going to read this plot from right to left. So in the one condition, it means that we have the original synaptic strength of this inhibitory loop. And as we constantly decrease the strength of this loop, eventually we killed the gamma oscillation because they fall below the threshold of statistical condition. And very interestingly, we found instead that beta increases, as we have already seen in the previous slide. So meaning that gamma oscillations needs in the globus pallidus the self-inhibition. And interestingly, self-inhibition is counteractive as with the beta oscillation, because when we kill, we also observe an increase in the power. So we have seen that the self-inhibitory loop is necessary for gamma oscillation, and then we wonder whether it is sufficient. By sufficient, we were interested in addressing whether these gamma oscillations comes from some properties of the inputs of these nuclei, or whether this nuclei alone was sufficient on its own due to resonant properties of the self-inhibitory loops to display gamma oscillations. And to do so, we basically took the very same model, the very same nucleus, and we substituted its input with Poissonian distributed inputs of same average level, meaning that eventually we feed the same net energy, but we completely destroyed the spectral properties of its input. And what we found is that, of course, in the healthy condition, you can see that there is no difference in gamma, suggesting that this loop alone does not inherit the gamma oscillation from its input, but can sustain. And another observation we can see is that these experiments killed beta, because you may remember that actually beta comes from the interplay of different nuclei. So now we are substituting them with Poissonian inputs, and it is killed. In the Parkinsonian condition, instead, of course, in the original scenario, we see the increase in the beta oscillations, and this is something expected as a sanity check. But what we found is that gamma oscillation can be displayed by the nucleus, but we were not that satisfied between the two feet. And this can also clearly be seen here, that for example, take a look at the central frequency of the gamma oscillation switching from the LT to the Parkinson condition. In blue, you see the original nuclei that has this non-monotonic behavior, when instead in gray, the isolated scenario, the one in which we feed with the Poissonian inputs, meaning that there is some difference between the two scenarios. So the gamma oscillation can be generated from the self-inhibition in the globus pallidus, but not quite the exact of what we observe in the scenario in which we have all the nuclei actually connected. And the difference, in my opinion, can be clearly appreciated when we take a look at the raster plot, because what's going on when we have a nucleus in the isolation and interconnected nuclei? As I said, we have the emergence of beta oscillations, which comes from the interplay of different nuclei. And you can see that on the left, on blue, you have the beta oscillating cycles of the firing rate of this nucleus. And instead, here you have the scenario in which we have substituted it with the Poissonian inputs. You can see that on average, they display similar behavior, but what's going on when you have the beta oscillating cycles is that the beta oscillations pushes this nucleus and gives it energy to display higher firing rate, and therefore, if the gamma oscillations is a function of self-inhibition, a higher firing rate is going to entail a higher firing dynamics and a higher self-inhibition, and eventually higher and faster gamma. And so we did also some more slightly sophisticated analysis in which we investigated the phase amplitude coupling when we have the beta oscillation with the power and the peak frequency of gamma, meaning that these self-inhibitory loops can sustain a globus pallidus gamma oscillation, but the presence of beta do interact with it. It gives another set of energy to the input to the globus pallidus in order to display faster and more powerful gamma, meaning that the two loops eventually interact. Then, so it is, sorry, you can really see, but we investigated for the globus pallidus, for this nucleus, the necessary and sufficient condition, and we found that self-inhibition of this nucleus can reproduce and sustain a gamma oscillation. The point is that when we have a beta oscillation in the Parkinsonian state, the two interact. Then we did very basically the same for the other type of gamma oscillation that we saw in the other nucleus, so the striatum. Now we're going to focus on the Parkinsonian condition, because in the healthy condition we did not display the gamma oscillations, sorry. And so again, we're looking for the necessary condition. Unsurprisingly, we found that self-inhibition, this time within D2, is the synaptic connection necessary to reproduce gamma oscillation. And you can see on orange, we have the original firing rate spectral density of the nucleus, and in blue, what happens when we cut these self-inhibitory loops. And we have, again, the disappearance of the gamma oscillation and the emergence of a more powerful beta oscillation. And again, we did also in this continuum from 1, in which we have the original scenario, until 0, in which we completely destroy this self-inhibitory loop. And we see a similar pattern of what we observed before. We have beta oscillation that's increasing, meaning that the self-inhibitory loops counteracts beta hypersynchronicity, when instead we have the gamma oscillation decreasing so much so that eventually in the halfway of the destroyment of this self-inhibitory loop, we have the gamma oscillation falling below the threshold of statistical significance. So we did the necessary condition, now we're looking for the sufficient one. Is this self-inhibitory loop on its own able to display gamma oscillation? For the globus pallidus, we found that yes, it was sufficient. For the striatal, instead, no, it was not sufficient. Because take a look at what happens in, first of all, at the beta oscillation, going from healthy to Parkinson's disease, and you can see that as a sanity check, it increases the beta oscillation. In the original scenario, you can see that, again, we have a healthy scenario in which we do not have striatal gamma oscillation, but then halfway through from healthy to Parkinsonian condition, we have the emergence of gamma oscillation. But when we substitute these input to the striatal with the Passonian equivalent inputs, we have, again, an increase in gamma oscillation, but they dance around eventually around the threshold of statistical significance, meaning that on its own, this nuclei is not able to reproduce gamma oscillation we observe in the whole network. And again, the reason why, what happens between Parkinson and healthy condition is the presence of beta. So, very long story short, this striatal population on its own does not have the sufficient energy to produce a self-inhibitory loops that eventually lead to an emergence of gamma oscillation. But when instead you introduce a beta oscillating cycle in the firing dynamics, you transiently feed sufficient energy to the system, to the striatum, in order to, in these very short windows, have a sufficiently high firing dynamics to trigger a self-inhibitory loops that eventually triggers also gamma oscillations. And this is the reason that when we have beta oscillation in this striatal population, we do have also gamma oscillating patterns. So here instead we said that, again, self-inhibitory loops is a necessary condition for gamma oscillation in the basal ganglia, in striatal gamma oscillation, but instead is not sufficient because this loop on its own has a low firing rate that cannot sustain the emergence of gamma oscillation. But when introducing the beta oscillating cycle, we feed to this nuclei sufficient energy to display gamma oscillation. So this is what just said. So we're already at take-home message. We have two different distinct gamma oscillations, one in the striatum and one in the globus pallidus. They have a common rhythmogenic, the mechanism behind the rhythmogenesis of these two nuclei is common, that is self-inhibitory loops. And we have a huge difference between so beta and gamma oscillation. Beta emerges, the beta hypersynchronicity in the dopamine-depleted Parkinsonian state emerges from the interaction among different nuclei, and we see two different subloops. Instead, gamma oscillations comes from what is usually being called the ING mechanism, meaning that it is local. It comes from local interaction within globus pallidus and striatal nuclei, which is exactly the opposite of the widespread nature of beta hypersynchronicity. And but the two are not in isolation. The two interact. The presence of beta oscillating patterns of excitability within the basal ganglia, as we have seen, impacts the emergence and the features of gamma oscillating patterns. And this aligns with recent experimental evidence in which you can actually see, especially in rat model of Parkinson's disease, but only in human, the presence of gamma oscillating patterns within the basal ganglia. And they're actually being currently investigated as an additional set of biomarkers to drive adaptive deep brain stimulation technologies. So future works, as we've seen so far, the inputs from a Gaussian speak, the inputs actually matters a lot. So as of now, we are just feeding Poisson-like energy to the system, but we're going to take into account a more realistic drive, especially from the cortical inputs that as of now is just contact, but actually matters a lot. And we are currently working on actually taking something more realistic out of our simulation in terms of the eventual outcome, because as of now we're talking about spectral firing dynamics, but we would like to include a proxy in order to estimate local field potential out of this computational model. The point is that we're getting working hypothesis or preliminary results that suggest that local field potential in the subdynamics nucleus or in general in the basal ganglia obeys to different rules as the one that usually applies in the cortex. So last slide is this us. This is the computational neuroengineering laboratory. On the top left at our most presentable. On the bottom right, what you usually see if you come in the lab. And these are the guys, the brains responsible for these work. There's actually also Professor Cataldo from Department of Physics at University of Pisa, but I did not have a picture with him, unfortunately. And this is our paper interview if you want to find out more, and thank you.
Thanks, Nicolò. Questions? Please.
Would you consider looking at other simulators that affect dopamine, like Huntington's? Or aside from degenerative ones, would you also consider looking at like dysregulated dopamine pathways, schizophrenia or?
Sure, it could be, of course, interesting. The point is, as of now, our dopamine depletion, the way in which we model dopamine depletion is very Parkinson's specific to some extent, meaning that in Parkinson's you see imbalance between the two striatal subpopulation, D1 and D2. So other models of dopamine depletion, or if dopamine depletion eventually leads to dynamical changes that differ from it, you would need more sophisticated modeling to take into account also other pathologies. So I'm not claiming that this model can travel well along all the other possible dopamine depleted conditions, but in principle, once you get a decent model of what's going on when you are in the dopamine depleted state, you might eventually try to use it as a benchmark in order to try to investigate the rhythmogenesis beyond other pathological states. But of course, you need to have first a good intuition of what's going on when actually dopamine depletion happens in other pathological conditions.
Okay, other questions, please.
I said I'm really associated with the session after this, but your title suggested to me an allusion to Aldous Huxley's Brave New World. What happens when the beta and the gamma get together and they have different rhythms?
Yeah, indeed, because eventually we were fascinated about the fact that the two are not completely independent, but actually they do interact with one another. We were not expecting that. We were expecting because the two have actually two different mechanisms eventually of rhythmogenesis. So eventually we were expecting not much crosstalk, but eventually we found that I was too tempted to put that title.
Other questions?
No, sorry, I can't hear you.
So, as a more broad question, in the group you do a lot of translational work and you try to link indeed these models to the actual activity or neuroactivity of patients. Do you think that the introduction of this loop might help distinguish better between the variety of pathology of patient pathologies, so to distinguish more?
Well,
because you introduce another.
Yeah. There are several points because it depends on what we are talking about when we talk about the signal you can collect from the patients, if I understood correctly your question. So as of now, our model can be applied to what you're suggesting if we get to know exactly the firing of each neuron within the network. Point is, we do not know. I mean, in computational model, of course we can. And as I was suggesting, if I understood correctly your question, we would need also working hypothesis in order to link the output of our modeling efforts with something you actually pick up experimentally that is not that informative as much as we do in our computational modeling. So as of now, we are working on it, but a link between what you observe experimentally and the output of our modeling is currently a work in progress. If I understood correctly your question, because I couldn't hear all of it. Okay.
Okay, maybe you can talk about it in the coffee break that now we start, thanking the session.